to present the results from a phase 1, phase 2 study of EV06 ophthalmic solution for the treatment of presbyopia. I want to say up front, in case I run out of time at the back end, it works to restore bilateral near vision. It's safe, well tolerated, and we're not here to try and raise money. We just finished a successful capital raise. Those we entered into our study between ages 45 and 55 represent a total of 47 million people in the US. We all know the market is big, and it's reassuring to know that we've got something coming that's really quite different in a treatment of presbyopia. So what is EV06 and how does it work? Our target is the lens. Let's see if we can't restore natural function to the lens as it becomes too stiff to change shape during a near vision stimulus. You've heard the fluid that moves from the side of the lens to the center of the lens, then back out again in accommodation and disaccommodation. That's where our focus is for treatment, and let's drill down to where that fluid actually moves is in the lens fiber cell, from the periphery to the center out back. And in a young lens, that works pretty good, but in an older lens, the events of oxidation strip hydrogen atoms from the sulfur atoms that are attached to the crystalline proteins. And then the crystalline proteins with the sulfur atoms to satisfy a charge join together, and these huge molecular weight crystalline proteins start to compromise the flow of fluid in the lens fiber cells back and forth, and this continues year after year after year of the disulfide bonding, and it just compromises the flow to the point where near vision is compromised and grinds to a halt. So we are administering in our clinical study a prodrug, well protected by patents. It's a choline ester of lipoic acid. When applied in a drop to the eye, the ester bond is quickly hydrolyzed, and we have choline and lipoic acid. Choline is needed by all cells, essential for life. Lipoic acid is mitochondrial in origin. It's diffused through the aqueous, actively taken up into the lens, and further reduced there to dihydrolipoic acid, which, if you can see, then can contribute the hydrogen atoms after reducing the charge between the sulfurs, and we restore a more fluid component to the cytosol in the lens fiber cells and restore near vision. So we have conducted a clinical trial. It was in two parts, a phase 1 part, a phase 2 part. Phase 1 part, enroll subjects and treat them for a week and make sure the medicine's well tolerated twice a day. Start with a non-dominant eye, and then at the end, when we're assured that the medicine is well tolerated, begin bilateral treatment. And you can see the subjects that we enrolled had to have best distance-corrected near visual acuity of 20/40 or worse, and a 20/20 best-corrected distance vision. There's some demographics from the study, 4 sites, all in the US. Mean age was 50.5 years. You can see it was predominantly female, predominantly emmetrope, and predominantly Caucasian in our study. No subjects dropped out. It was well tolerated. Comfort was good. There was no change in distance-corrected near visual acuity, and there was no change in pupil diameter through the study. We achieved both of our primary efficacy results in terms of distance-corrected near visual acuity throughout the study, which continued, and the proportion of subjects with greater than 10 letters of improvement in distance-corrected near visual acuity. So here in a logbar scale, you can see the placebo in the green line. The blue line is EV06. Nice p-values. There is a placebo learning effect with presbyopes that I think most people know about, but as you can see with the blue line, it continued in a really great direction throughout the study. So what's this look like translated to Snellen? You can see day 1 is the curve on the right, and day 91 is the curve on the left. 82% of the subjects had 20/40 or better vision. 20/40 is not bad near vision. Great improvements throughout. The placebo group, not so much. Not much of a shift. So it restores near vision. Additional studies warranted. We're going to follow subjects at 120 days and 270 days to see the duration of the treatment effect. I want to thank the clinical sites, and certainly want to thank the Encore Vision team, its advisors, and board of directors. Thank you.
Bye.